We're going to finish up talking about single-celled organisms known as protozoa. Now, in many classification systems, protozoa are a separate kingdom known as Protista. And in the classification system that we're using, this is a separate kingdom. Protozoa are all microscopic and represent approximately 30,000 living species. One group of protozoans is known as the flagellates, and we've been exposed to the idea of flagella before, flagella being these long whip-like structures that are used for locomotion. And the prototype of the flagellate is the euglenia, which is pictured here. The euglenia has the long whip-like structure known as the flagella, which is this area right here. And then this is another diagram that I would strongly recommend that you take a look at. The front of the organism where the flagella is where the eye spot is, the light-sensitive area, a gullet or opening where food enters, and a reservoir for that before it is passed into the rest of the body. There are contractile vacuoles, which are used to expel water. And then the interesting aspect of euglenia is they also are the only organism or the group of organisms that also have areas, a chloroplast, so they can do photosynthesis. And so you have the fact that they eat like animals from time to time, but they also can produce their own energy through photosynthesis. And so the pedicle is a thick membrane which acts as a cell wall to protect the euglenia. The movement of the euglenia it occurs by the use of the flagella, but also by the fact that the euglenia can change the shape of its body and lead to movement. The gullet is an area for the intake of material, and as I said, the contractile vacuole discharges water and liquid waste, much like a kidney. The nucleus of the euglenia is where the DNA occurs, and euglenia reproduce by conjugation, which means that two euglenia come up against each other and there's an exchange of material, and then there's production of a new euglenia. Chloroplasts contain chlorophyll, which participates in photosynthesis. The eye spot is a light-sensitive area, and euglenia tend to move toward light areas. And they, as I said, reproduce by conjunction and binary fission, which is an asexual reproduction. A second flagellate is called trypanosome, and it's transmitted to humans by the fly. And trypanosomes cause what's called African sleeping sickness by Trypanosoma cruzi. The fly actually lands on the individual, and when the fly defecates and leaves a small stool sample there, the person scratches, and then the person becomes infected by scratching that area. The trypanosome has what's known as an undulating membrane, which is attached to the flagellum and helps for the movement of the trypanosome through the blood. The next group are sarcodines, which are protozoas with pseudopods. We think of amoeba. When you see an amoeba or hear about an amoeba, this is the group, the sarcodines, is what the amoeba are. And here's a example of an amoeba. Very simple, with a nucleus, a contractile vacuole, which functions the same way it does in the previous euglenia, and food vacuoles. It takes in food by phagocytosis, and then the pseudopods, or false feet, is how it moves through the environment. The pseudopods, as I said, are extensions of the cytoplasm that are used for movement and to capture food. And food vacuoles are where the food is ultimately digested. And similar to the euglenia, the contractile vacuole, it regulates the water content in the cell and expels water and waste. The amoeba are important to humans because there are certain amoeba that can cause an intestinal disease, which is called amoebic dysentery. They can also result in abscesses in the liver. There's an amoebic liver abscess. And this is how an amoeba eats. You start here, it senses the food. It extends pseudopods out toward the food and engulfs the food. The food is then taken in and a food vacuole is formed. The food is digested with enzymes. Then what's left is expelled and it is ready to take in more food. Amoeba also can form form shells of calcium carbonate when there are serious problems with the environment. And this is called foraminiferanes. And these shells can be quite intricate and made of silica. And the organisms are called radiolarians. Now there's protozoa with cilia, and these are called ciliates. The cilia are hair-like projections which help with locomotion, and the classic ciliate is paramecia. This is the paramecium here, and I'll go quickly through this diagram and then we'll probably talk about it again. The oral groove is what takes in food. It then forms a food vacuole, which is where it's digested. When food is expelled, it's brought over to the anal pore and expelled through the anal pore. As I said, they move by means of the cilia, and then they have two nuclei, the micronucleus and the macronucleus, which we'll talk about in just a minute. They have a contractile vacuole, which functions very much like the contractile vacuoles that we've seen in previous slides. So the macronucleus is responsible for basic cell activities. So the large nucleus or macronucleus is the basic activities. The micronucleus functions in reproduction. The trichinocysts, which are shown here, right here, are dart-like structures that are used for defense. And as I said, the oral grooves are where food's taken in and the anal pore is where it's expelled. This diagram shows the conjugation that occurs between two paramecia. They attach to each other. There's an exchange of the macronucleus disappears and the micronucleus divides and is swapped. The paramecium then separates and then goes through binary fission and forms new paramecia. So before the division, you have a single paramecia, the macronucleus disappears, the micronucleus is reproduced, it elongates and divides, and then after division you have two. This is when it occurs without conjugation. This is with conjugation. So the protozoan with ciliates, there are three types of protozoan with ciliates shown in this slide. First, the duodenum, which is the central structure here. This is a barrel-shaped structure which eats paramecia. The vorticella, which is over on this end, is a stalk-like ciliate. And then here, on this end, is the stentor, which is a trumpet-shaped. The taxis of a protozoa, what does that mean? Well, taxis means the ways that they move. So chemotaxis is movement that's caused by chemicals or attractions. So they're attracted by chemicals and they move in the direction of certain chemicals. So that's chemotaxis. If you're attracted by light, it's phototaxis. If you're attracted by gravity, it's gravitaxis. And if you're attracted by electricity, it's electrotaxis or electrical fields. Sporozoa are protozoans that don't move. They all have two hosts. So their sporozoans always exist in two hosts. So for example, malaria, which causes a febrile disease, is caused by sporozoans. And I bet you can guess what the two hosts are. The mosquito and the human. Plasmodium is a specific cause of malaria, and it alternates generations between the two hosts. So there's one generation that forms in mosquitoes that's passed to the human and forms a separate generation, which is passed then to a mosquito, etc., back and forth. And this just shows an example of that. The infected mosquito bites the human, and the plasmodia is injected into the bloodstream. The plasmodia invades the liver cells and multiplies. Then the plasmodia exit the liver cells and attack the red blood cells. The red blood cells, after 48 hours, all of the red blood cells burst at the same time and release the plasmodia, which invade new blood cells. And then the same thing happens again, but gametes are then produced in the red blood cells, which are in the blood. And then when an uninfected mosquito bites, it takes in these infected red blood cells, and these gametes then combine to form a new plasmodium, which is then injected, and the beat goes on, if you will.